Hi everyone, welcome to another Fagron seminar. So today we have Raoul Wolf. He's a new member of the section for Ecotoxicology and Risk Assessment, and he started his postdoc around 3 months ago.
Yep.
And he's going to talk to us about his journey from ecotoxicology to ecology and showing some mortality during his PhD.
Yeah.
So the floor is yours.
Thanks for the intro. Yeah, so obviously after 3 months being at NIVA, there's not a lot I can show from what I've done thus far, but hopefully a bit more of what I have been doing before coming here. So kind of, it's almost like a personal recap of my journey from ecotox to ecology and data science and statistics, and what I've learned as an ecotoxicologist going through other disciplines and then coming back, and also what I think where other disciplines might profit from ecotoxicology. Yeah, this works pretty well. So first, a few words about where I come from, and you might be able to tell from the voice I come from Germany, southern Germany, that is. And in every talk like that, I just have to showcase the village where I grew up. I grew up precisely there. And it's a small village near Heidelberg. And as some of you might know, Heidelberg has has Europe's oldest university, I guess. And I took my bachelor's in biology there, focusing on ecology, ecotox, limnology, and other disciplines. But writing my bachelor thesis in Germany, the system is a bit different from here, so you also write a thesis for a bachelor on ecotoxicology in the group there, endocrine disruption in zebrafish. Later on, I went on to take a Master of Science in Frankfurt in environmental sciences at the university there, and broadening the scope a bit there, so going from ecology, ecotoxicology, but also including environmental chemistry and nutrient cycles, and then writing my master thesis on mixture toxicity in Daphnia. So both bachelor and master thesis, pretty much ecotox as ecotox gets. And then for my PhD, I actually went to the University of Oslo, not too far from here, where I worked mainly in actually ecology, but also ecotoxicology and a bit of limnology, modeling, and statistics. And my PhD was on the toxic effects of water browning on Daphne. And when you look at the sort of the Stippeuhr that I have here, wow, it's always strange when Norwegian comes first. And see where sort of the common denominators of these something, of course, is to make a word cloud because it's fancy. And there you see that a lot of what I have been doing is actually ecotox, limnology, ecology. And one thing I came across, or a principle I came across, if you want, is one of the things we can do is actually explain eco with tox. This is sort of what brings ecology and toxicology together in sort of my very limited brain space. And for that, we don't even have to go back that far. If we just look at examples of how ecotoxicology came about is, for example, you have in the 70s, 80s, you have certain spills, you're at a lake or at a river, and then you figure out, well, there is, let's just say, an environmental change, whatever that may be. And then you do some research and then try to figure out what the cause of this is. And well, yeah, maybe a chemical plant burned down upstream. And so you find the explanation for this. So it, in the very beginning, is a ecological phenomenon. There's a bunch of dead fish. And then you later on try to investigate and find actually a good explanation or a direct link, that is. And this is pretty much how ecotox came to be as a discipline. But as I hope most of you will agree, nothing in ecotoxicology makes sense except in the light of ecology. I always had the feeling that some of the ecological aspects are, say, a bit underrepresented in ecotoxicology. And so I tried to show you a few studies now where I hope I succeeded in linking the two a bit closer together. By the way, if there's any questions, just raise your arm, shout, whatever. So, explaining eco with tox, and this is now about my PhD that I took. So if you think of the world like this, you have a functioning ecosystem here. Imagine this here being a lake with a river with input, and then you have some houses, and then maybe you have some factories there, etc. But the important thing is the sun is shining into the water, and part of the solar spectrum is UV radiation. Now, most of you know pure UV radiation is pretty harmful to organisms. And just clearing that out here, imagine the same situation, but this time we have a new phenomenon called water browning. I won't get into the details of this, but freshwater bodies all over the Scandinavian area and also slowly in the Central European areas are getting browner, meaning there's more humic substances in it, more essentially dissolved organic matter, or short DOM. And what these additional molecules do is they essentially absorb harmful UV radiation, which also means there should be less UV radiation to actually damage organisms in the water. So that's good news. And this brings me to the first study that I want to show you, where we looked at genetic damages in Daphnia, the water flea, based on a 4x4 design, of a gradient of dissolved organic matter and a gradient of UV radiation. This is a picture of my Daphnia. Fun fact, the Daphnia were from Neva. Because our culture died back then. So just a quick word about the design. So you have a gradient in water browning, you have a gradient in UV intensity, and all sorts of combinations of this. And then we exposed Daphnia in there, took individual Daphnia out and performed an assay called the COMET assay, where you look at DNA damage. Those of you that know the COMET assay, you know that that can be pretty heavy lifting, so that took a long time. And when you invest so much time in sort of method development, in experimental work, etc., one of the things I felt sort of uncomfortable with is going with the regular data analysis in Ecotox, meaning, yeah, maybe a ANOVA, or you throw a t-test at it. And this little meme here, I think, pretty much exemplifies what happened to me. So I was walking along GraphPad Prism's ANOVA, and then the beauty of linear mixed effect models came across my path. And so I switched from any sort of point-and-click statistical software into what actually the ecologists use in R. And then I analyzed my data with this, and we were actually able to really find out some cool things. Just bear with me on this one. I know it's 3D and you're not supposed to show 3D things, but on the x-axis down here, you have a gradient in UV radiation. On the y-axis, you have a gradient in DOC, water browning, the higher the number, the browner the water. And on the z-axis, you have your DNA damage. You see three things here. First of all, the UV radiation that we used was not that strong anyway, so it doesn't really induce a lot of damage. It's sort of just borderline UV radiation. You see an effect of the organic matter alone, which I think is pretty cool. But the strongest effect that we see here is actually in the combination of the two, on the DNA damage of the Daphnia. And that's an interesting phenomenon that we try to find an explanation for. So we went back to the theory here. So what happens is that these molecules, they get hit by UV radiation, which turns them into an excited state, which causes the formation of reactive oxygen species. And reactive oxygen species that ecotoxicologists now have all sorts of bells ringing. Reactive oxygen species are, for example, known to induce DNA strand breaks, as the ones we measured there in our study. And then when we actually went back to the lab, did some in vitro studies to actually express the combinations of water browning and UV radiation as formation of reactive oxygen species. And then we were able to see some sort of good trend. I've seen worse, let's just call it like that. And that, I think, was a pretty exciting finding for us. So just to recap here, we found no damage from our UVA radiation, which was weak. So we had moderate damage from DOM, but the combination of the two caused the highest increase in DNA damage. And that is linked to the photochemically produced reactive oxygen species. So a question is, of course, well, is there a underestimated risk, or is this ROS formation something that's actually relevant in the environment? And if yes, where and how, and so on and so forth. And that brings us to the second study I actually did together with Erik here. Where we modeled this theoretical formation of reactive oxygen species from the interaction of the water browning molecules and the UV radiation for a data set we had on 77 lakes in Scandinavia. So actually lakes like this, and this is the location of the lakes, and you see it's sort of very well distributed in southern Norway and Sweden. And what we had for all of these lakes were absorption spectra for the DOM, sort of as a, how could you say, spectral property of the water browning. And we combined this with the solar photon flux, meaning actually what quality and what quantity of photons hits the Earth. And then we combined that with what's called the quantum yield, meaning how many sort of ROS molecules you get per photon at different wavelengths. And we modeled the ROS formation from this. And in this study, most of my work looked like this. So I sat there for weeks and weeks and weeks, waving my magical R wand this time, and tried to make sense of this. And this work actually has taught me a lot about how to organize your data, how could you say, create your graphs and statistical analysis in a way that you can comprehend the week after. If it might sound familiar to some, right? But back to the science aspect here, I was showing the solar photon flux here, and obviously the colors are sort of reflecting the visible light here, and UV radiation is down here, essentially between 300, 320, and 400 nanometers. And you can see just by the sheer amount here, it's not a lot of UV radiation that actually reaches the surface. And by the way, does anyone know why at 400 nanometer wavelength there is a lot more photons? Yeah, it's the ozone layer, correct, which is absorbing all the UV molecules. And that in itself I found quite interesting to see, okay, so we are constantly worried about UV being also evolutionary driver, skin cancer, yada, yada. But that's actually so a minuscule proportion of the photons that hit the surface of the Earth are actually UV. And just keep in mind that at 400 nanometer, we see a sharp increase in the number of photons. And so what we modeled here is the theoretical Ross formation in these 77 lakes at the very surface along the wavelength of the light. And we see here from 300 to 400 nanometer, we have like a peak of Ross formation, meaning at the very surface, UV light causes a lot of Ross formation. But then we also have like at 400 nanometer, we see another peak. And remember the photon flux I showed you where sort of at 400 nanometers, we just have more photons. They contain less energy, but there's just too many of them. So that you see this effect. And if we go 1 meter deep into the water column, again, 77 lakes, you see this here. Actually, the Ross formation at a depth of 1 meter is not really dependent on the UV radiation anymore, because most of it is absorbed. But then at 400 nanometer in the visible light, you just have more photons passing through the water column, actually being there. So even though they are less efficient in producing Ross, at least they are there to produce Ross, right? And this also allowed us to create a profile of Ross formation throughout the water column, from the surface down to, I think the deepest lake was around 70-something meters, and logarithmic scale on the y-axis here. And you see that the majority of the Ross production actually takes place sort of, if we're very nice, we say within the first meter of the water column. You could also argue that the majority takes place in the first few centimeters of the water column. And now trying to link up this information here with the information we had from the previous study where we looked at DNA damage from Ross formation. Of course, it would be cool to know, like, okay, depending on the depth, what type of DNA damage could we expect? And this is what you see here again. So on the, just let me walk you through here. On the x-axis, you have Ross formation from the lab study and DNA damage. On the y-axis, this was the Daphnia experiment, study 1, and the red line is sort of our regression here. And the blue stripes that I hope you see are the modeled Ross formations in these 77 lakes during a day. So in the first centimeter of the water column, you roughly have a little less than, what is it, 200 micromole per liter Ross formation. And the deeper you go, the more minuscule the Ross formation gets. So we see that actually within the first, say, few centimeters, we don't expect everything to be super damaged just from Ross, but we see that it's shortly before the effect kicks in, which then brought us to a third study I'm going to show you, where we looked at the behavioral response of zooplankton to UVR in the presence of water browning. This is within the first few centimeters of the water column here, which is what we were interested in. So the cool thing we did here is and this is the setup that we used. This is no joke. So we used Lego and whatever we had gradients in DOM and UV light and non-UV light and everything. And then we thought, well, instead of spending so much cost or time or whatever and buying some stuff, we built our own robot to actually go there and take the movies. And what happened is whenever you try to save time, you have to hide the pain. It took a lot of time, a lot of troubleshooting, etc. But we managed to actually get it done. So again, we had a robot that was essentially just going from left to right, just going from left to right. difficult enough, with a camera on top, and then filming different bottles with one Daphnia in each. And as you can see here, there was a gradient, a randomized gradient in water browning, and this was either with or without UV being present. And then we analyzed the swimming behavior later on, which also taught me a lot on how to actually handle big data, because if you take videos, you quickly end up with gigabytes of data for these experiments. And one of the results here, which is kind of a proof of concept that we actually didn't mess up too much at least, is here you have very quickly on the x-axis the exposure time, because we ran these experiments for 6 hours and they were filmed sort of in intervals. And left side without UV, right side with UV, and the different red brownish colors indicate a gradient. And on the y-axis you have the swimming depth, which of course at 0 above here would be the surface, and here at 40 millimeter per centimeter, this dashed line is actually the bottom of the flask. So they couldn't go any deeper than that. And the basic thing we see here is, well, without UV, they are pretty much in the same areas. With UV, they are much deeper, but the browner the water is with UV, they get a little higher. So there we see that it's a small effect, but it is a statistically significant effect that water browning buffers the flight reaction. Yeah. Yeah. That's true. We ran pilots on this because we simply didn't know how to sort of measure this in everything. And what we did is we had lots of fans that kept the temperature constant, at least plus minus 0.5 degrees. So we ran into the trouble earlier on when sort of prototyping this setup that after 6 hours, I think some of the Daphnia were dead because it was getting close to 35 degrees or something. And so we just took fans and increased the distance a bit. And that worked quite well for us. But yeah, it's a good point. This is something we also in the beginning struggled with, because obviously we thought, well, must be sort of the UV radiation or something like that that's killing the Daphnia, but then you see them dead without UV as well, and then you start worrying. Yeah. But in this analysis here, we didn't have a way to sort of account for the heating there. So we tried our best to minimize the impact of this heating, if that answers the question. So this was swimming depth. And then we also looked at the swimming time, because, you know, Daphnia, they swim a bit and then they are sort of static, then they swim again, then they remain static, then they swim again. You get the point. And then we looked at this in the presence of UV and without the presence of UV. And again, this gradient in water browning. And the interesting thing we see here, where I don't really have an explanation for this, but it's there, is that the activity, the swimming activity of the Daphnia increases with increased water browning. Irrespective whether or not we have UV or not, sort of this water browning effect is still there. Yeah, so to recap here, water browning buffered the behavioral responses of zooplankton, which was probably due to the absorbance, and water browning also increased the overall swimming activity. And now we'll just quickly jump over this summary, because we're slowly running out of time, and quickly present this sort of from everything I did before my PhD to what I did in the PhD to me standing here now is somewhat sum up here that I have a core skill set here with ecotox, environmental chemistry, ecology, zoology. But then I personally profited from actually stepping outside the comfort zone, talking with ecology people, limnologists. Did I profit from talking to statisticians? That's the question I leave hanging in the air. Yeah, of course I did, but with the modeling as well as omics. And I've been involved in field work, lab work, data work, if you want. Some of you might have seen we've announced a workshop on how to use R for reproducible scientific analysis. Next week, in 2 weeks, the course is fully booked, but there may be something happening internally, just to give that away. Which brings me to what I actually do at NIVA. RAW are my initials, which I think is kind of cool. It's at least my best email address I had thus far. I have a 3-year postdoc here. I'm 75% on a project called Mixed Risk, which I will say a few words in the next slide, and 25% other projects, Pflichtarbeit. And at Mixed Risk, what I'm currently working on is I'm working on different work packages here. And the thing I'm working on most now is actually the development or improvement of the risk assessment database. If you are unsure what that is, what that means, etc., I've been told that my project leader, Knut Erik, will give a FARC lunch later this year. And he probably has a lot more to say about this than me after 3 months, mind you. So what we do here is we work on a risk assessment database, and then the idea is to later go from in silico, meaning like modeled results, try to get a proof of concept with in vitro or in vivo studies, and also not the least to employ some more modern statistical methods in risk assessment, because in ecotox traditionally, if it's sort of beyond the scope of ANOVA and t-test, it's voodoo. But that's a lesson I took from my PhD, that it's actually, well, it still remains voodoo, but you see, it's friendly voodoo, to put it like that, right? So what I currently work on is try to make sense of 2.5 million chemicals and information on these chemicals from different data sources, try to sort of get them together and get some properties for these chemicals from several APIs, meaning that I have a website and then I know, hey, for this compound, could you give me, say, the molecular weight? But obviously you have to automate that because you are not copy and pasting into a website 2.5 million times. And another thing I work on now is trying to make sense of organism taxonomy, which is giving me a bit of headache. Again, with 2.5 million different organism ranks. So that's still from one data source, but we may have to sort of include others. But when you work with organisms and names, it quickly gets difficult because even the scientific name might have changed, might have synonyms, etc., etc. We even have data from animals that are extinct now. It's quite strange. And I work mainly now on the Oracle PL/SQL server here, and do most of the sort of data tidying, merging, prototyping, etc. in R. And so the final slide of this is the sort of expanding mind. I mean, here, how you go from ecotox to ecology to statistics and data, but the real pleasure happens when you go back to ecotox. And yeah, with that, I commence my talk pretty much on time. Thank you.
Thank you very much for your nice presentation. Unfortunately, we don't have time for questions, but I guess you can always
No, please,
Talk
it's,
with Raoul
yeah.
afterwards.
I'm available for questions every time. Thank you.
Thank you.